And now I have the pleasure to introduce our keynote speakers on there. And we will start with our first guest and speaker today, Elisabeth Janssen, and from the Eawag. I would like to briefly introduce her to you. So she started her studies in Germany at the University of Duisburg in studying environmental science, and then moved to the US, continued the masters and PhDs in Stanford, again, or continuing environmental science and engineering. Did a postdoc in the Natural History Museum in London, and another postdoc in environmental photochemistry, where probably the light came into the whole business on there at ETH Zurich on there. Her research interest is on determining the chemical information that is in water. And this is, of course, a topic of highest importance, because clean and healthy water is essential for everybody of us. And light has an enormous impact on water. And as a chemist working with light, sometimes even using water as a reaction medium, I know that is a very, very important topic. So without further ado, Elisabeth, Lizzie, thank you for being here. Go ahead. We're looking forward to your presentation.
Well, thank you very much. I'm excited to be part of this and learn about all these other disciplinary ways to look at light. So I'm also want to thank the organizing committee to have me share or give an overview of what we think of aquatic chemistry and sunlight. So when we think of light in the surface water, in my case, to facilitate my studies, there are two aspects I'm interested in. So I want to see how light helps life, so all the organisms depending on the sunlight and water, how that is happening. And then at the same time, what we do very much is to understand how light can help us degrade pollutants and chemicals that we don't want in the water. And I give you a couple examples, like just how we study exactly this. So starting with the sunlight spectrum, we have these different energies of light, of course, and as soon as the light hits the water surface, different parts of the spectrum are able to penetrate to different extent into the water column. So we know, the infrared light warming our surface water layer because it gets absorbed mostly in the surface water. But we have these higher energy portions of the sunlight that can penetrate much deeper into the water. And for us, this is the really an interesting part, because this is the light that can help life evolve in the water or grow in the water in the first place. And also this is the portion in the water where light and pollutants exist at the same time. And we can understand how photochemistry can also support degrading those unwanted pollutants. So we study the sunlight, not always directly in the surface water, just because it's just not as reproducible at times. We do put our experiments directly in the sunlight, but mostly we use special light reactors. So we simulate the sunlight. So we're just independent from the season or the place on the planet we want to study sunlight. We have these special reactors where we can use just a certain portion of the sunlight spectrum that is relevant for our chemical reaction. As all the way down to going to use laser light, where you really can take a very fine slice of the sunlight spectrum and control in your experiment that only the chemical of interest is reacting, and you don't have as much side reactions that you would otherwise observe. To give you an example, when we study the pollutant degradation, of course, we can study pesticides, industrial chemicals, pharmaceuticals, and so on. Basically everything that we are concerned about in terms of water quality. And one example I want to just quickly highlight is, for example, studying pain medication like Diclofenac. This is coming in to our surface water because it's not completely removed during wastewater treatment. It's a pain medication we probably all know. It's pretty ubiquitously used throughout the population. That's why we can find it in almost all the surface waters that receive treated wastewater. And here it's very nice to understand how much sunlight helps to decrease the concentration of pharmaceuticals like this in the surface water. So Diclofenac can absorb part of this really high energy light in the sunlight spectrum. So everywhere where that light reaches, we can understand and model how fast diclofenac is degrading. So we can, for example, study diclofenac in a group of very similar pharmaceuticals. And here you basically just see how the concentration is decreasing over exposure time in the sunlight. Each line is a chemical. And then the steepest line here is diclofenac, which is degrading the fastest in this case. And what we then really do is we go into much more chemical detail to understand the reaction mechanism behind it. That helps us also identify and find reaction products to learn if they are of more or less problem than the original compound. But most importantly, we derive parameters that are specific to these reactions in light and that are system independent. So then we can go on and predict for other chemicals and other systems how fast this sunlight degradation might occur. Besides studying these, what we call micropollutants, another part of my work focuses on enzymes in water. So here, the relevance is basically that these enzymes are for microorganisms what knives on the plate are for us. So we use our knife to cut our food, to put into our mouth. And enzyme or microorganisms in surface waters, they can send out aquatic enzymes if they need to get to nutrient like food sources that are too large outside of the cell to be taken up. So the enzymes chop up the organic matter outside the cell, and then the organisms have bioavailable food. But once these enzymes are outside, they are extracellular and they are just very vulnerable to be degraded. And one way, of course, is again being degraded by sunlight. So we really want to study what happens to this enzyme in the sunlight, how fast is it inactivated? So we can study, it's a pretty big molecule, but we can study each individual part of this enzyme, and then in this heat map kind of representing where the most damage accumulates, and try to relate that back to why this enzyme stops working in the water, why it is inactivated. And because it degrades organic matter, it plays a very important role in nutrient and carbon cycling in the water. And the sunlight can be, in this case, put a stop and end to this activity. What I want to tell you a bit more about today is the third part, which is our work on cyanobacteria. And cyanobacteria are a living organism depending on life, but also causes several problems that we are investigating in the sunlit surface waters. So first of all, it starts out to be a good story because cyanobacteria were very fascinating masters of life. So some 2.5 billion years ago, when there was no oxygen on the planet, those guys came around and they basically changed that. They used sunlight to produce biomass, and in this process, they generate and release oxygen. So that sounds all good, but they have very specific traits, also fixing carbon dioxide, fixing nitrogen, that really gives them a growth advantage, that at times when they really multiply rapidly, we can have a situation where cyanobacteria temporarily take over a whole water body, meaning that they are the main biomass in that whole system. And then we start to have problems. And to illustrate this a bit further, what we call this a bloom, when we have this really dense situations, this is an example in the Baltic Sea. So this is a bloom of cyanobacteria that is 200,000 square kilometers in size. And as a reference, there's a line pointing to a ship close by. And the problem is, while the cyanobacteria are there and growing, for once they shield the light that would otherwise organisms depend on in deeper water layers. And while they are living, yes, they produce oxygen, but once this whole biomass has to decay, eventually when the organisms die, you have the opposite, that the biomass degrading consumes all the oxygen and you have dead zones. And if you think of the size of these blooms, this is a big problem again for the life in these areas. And then cyanobacteria are also not a simple single type of organism. They come in all different shapes and colors. There are many different, adapted to different conditions in that case. They are a global issue, not just in the Baltic Sea. We find them in many drinking water reservoirs in the United States and China. Often people there don't have an alternative freshwater source. So they sometimes are faced during blooms with water that looks like this, that they have to try and treat for drinking water. Here in Switzerland, we have not a pressing problem for our water quality, but we do have arguably in every surface water, we have cyanobacteria, but you know, like bacteria, sometimes they are good. The problem is when there are too many, like if we have an infection. So the seed is here. We just want to understand the cyanobacteria better, that they don't start blooming, or that we can control or manage our waters better, that we don't run into these significant problems. So sometimes we see in Lake Zurich temporary local blooms of this red blooming cyanobacteria. You may have heard of the incidences in Lake Neuchâtel last year, where a couple dogs died really quickly after being exposed to a very local bloom in the lake. And in other lakes in Switzerland, this happened this summer again. And if we hear these stories, then we really wonder, like, well, what is now the problem with the dogs? They don't mind the oxygen depletion or whatever we talked about before. This is the second part, and where my research again comes in, is that the cyanobacteria also produce toxins when they grow in sunlight. So the most famous toxin is microcystin LR that got discovered because of a tragic case in Brazil, where 52 dialysis patients passed away because they were temporarily treated with an alternative freshwater source that was trucked into the hospital. And later the investigation showed that this water was contaminated with cyanobacteria. They produced microcystin, these toxins that are hepatotoxic, causing liver failure in the patients. So very frantically, research was going on those couple of years, and then the WHO could put threshold values out there for drinking water guidelines. That was in the late 1990s. Today, we have 3 more toxins of cyanobacteria on the list, but notice that this took 30 years. So you need a lot of research evidence to come up with threshold values for potential toxins. And when I started this work, I quickly noticed that in the literature, there's a legion of bioactive compounds besides these few toxins that are in the WHO guidelines that cyanobacteria produce. And we have an open access database created recently where there are more than 2,000 compounds, and they're just not well studied. So we need to know, when are they being produced, when cyanobacteria grow in the sunlight, and how stable are they, how toxic are they, so forth. So with this list, we can at least start investigating lakes. So we monitor Swiss lakes. We take samples at specific depths to see how the cyanobacteria evolve in the season, and how these toxins are, these toxic fingerprints are looking like. So here's an example from May to October. And each color would represent a different compound. And you can see that the composition of these compounds is changing. It's not just one toxin being present, it's more like a cocktail of compounds being released from these cyanobacteria in a single lake. And then the last part is that we want to study how stable are these compounds in the sunlight, because we are interested in photochemistry of pollutants in general. So we can study these green forming cyanobacteria microcystis, or the red forming ones. Those are Plantotrix. Again, we see they produce a list of different metabolites, and the metabolites between species are different. They produce different amounts of each metabolite. And this is just a quick example. The orange sliver of this bar chart is the portion that would degrade in 3 hours in the sunlight. Can be something like 20% or as little as 6%. So we are very interested in our research to learn, why are some compounds degrading, and how is this actually happening? Why are some compounds not degrading? What does this mean for exposure of these chemicals, and if they reach drinking water treatment plants, etc. Unfortunately, I'm not going into much more detail. I hope this gives you a bit of an overview. So on the cyanobacteria side, there are many other projects we are involved in where light plays a different role. We study these fate processes that I talked about. We also study cyanobacteria in special communities growing on large areas on the rocks in the Swiss Alps and the mountains. We study how water treatment of drinking water decreases toxin concentrations using UV light and ozonation. And I collaborate with my colleagues in microbiology and ecology, because really understanding how these blooms form and how we can control this is a very interdisciplinary topic. And these are just current projects that are going on. So in the light of time, I'm thanking the funding agencies that contributed to the projects that I highlighted, and the students and researchers I worked with. And yeah, maybe we have some discussion also later in the coffee break together. Thank you.